Hello everyone. In this video, I will show you how to use the Flowmi cell strainers. These strainers are quite effective to refine the single cell or nuclei samples when using them with Tennis Genomics kit for single cell RNA-Seq, attack-seq or multiome assays. For such assays, when the sample is coming from primary tissues, it contains a lot of debris which cannot be completely removed with density gradient solution. Also, when isolating the nuclei, debris is again generated during the cell lysis step and it can often lead to clumping of nuclei. Therefore, it is essential to remove all the debris from the single cell or nuclear suspension before loading them into a microfluidic chip. If allowed to persist, then the debris will clog the microfluidic channels and will result in irreversible loss of sample. So these Flowmi cell strainers, they are 40 micron in size and they have a very small void volume of approximately 100 microliters and they are very easy to use. Here I have my nuclei from a primary tissue sample in 1 ml volume. First, I will take them out in a pipette tip. Here, I am using a low retention filter tip from Tarson's and for the nuclei sample. Thereafter, the tip has to be pressed over the strainer to firmly secure it. At this stage, we have to ensure that the pipette tip is held perpendicular while attaching to the strainer. If pressed at a slightly oblique or tilted angle, then a nuclei can leak from the top of the strainer. You can also apply some gentle pressure with your fingers. I am moving it a bit to firmly anchor it. These strainers, they seem to have minute teeth made of some metal to facilitate their attachment to the tip. Now I am ready to strain my nuclei. I have a fresh tube into which I will transfer these nuclei. So for this, I have to insert the strainer halfway into the tube and then by applying some gentle pressure at a constant rate, I am releasing the nuclei from the side of the strainers. So here we have to take care that we are not applying too much of the pressure, otherwise the nuclei can leak from the top of the strainer without being sieved and the sample can be again adulterated with the debris. I'm almost halfway through. That's it. I'm done with the straining. So these filters, they have several advantages that with their small size, their void volume is also very small. This helps to minimize the loss of the precious sample when we are dealing with low cell number, especially from primary tissues. They also help to reduce the aggregate arising due to clumping of nuclei or cells to some extent. Plus, the strainers will fit to the tips from almost any brand. The entire process is very easy to follow and consume very little time to strain the samples. Now the debris-free single nuclei suspension is ready to spin and can be again resuspended in a small volume. I will later examine them under a microscope before proceeding with the single nuclei multiome assay. Thank you for watching this video and I hope you find it useful for planning your next single nuclei multiome assay.